Welcome to AP Biology. This video is on enzymes. An enzyme is a protein that works as a biological catalyst. A catalyst is any type of chemical agent that speeds up the rate of a chemical reaction but doesn't actually get consumed in the reaction itself. So we will see that enzymes can be recycled. They can be used over and over and over again because they don't get used up during chemical reactions. All enzymes are catalysts. All enzymes speed up chemical reactions, but not all catalysts are enzymes. There are other things that have the ability to speed up chemical reactions as well. Here we see an example of an enzyme catalyzing a chemical reaction. The chemical reaction we see in this diagram here is the breakdown of sucrose through hydrolysis. Well, what's actually performing the process of hydrolysis? It's an enzyme called sucrase. You could always identify an enzyme because they usually end with ASE. Sucrase is the enzyme that specifically hydrolyzes sucrose or breaks it down into glucose and fructose. This brings up a good point. Enzymes are extremely specific for the substrate they act on. The substrate is another word for the reactant. So the substrate for sucrase is sucrose. Here we see an example showing how enzymes are recycled during chemical reactions. You can see that in this reaction the substrate enters the enzyme. While the substrate is in the enzyme, the chemical reaction is performed and the products are going to leave. The important thing to notice in this diagram here is that that enzyme is then available to perform more chemical reactions afterward. So these enzymes are recycled. They don't get used up in chemical reactions. How do enzymes actually catalyze chemical reactions? They reduce something called the activation energy. The activation energy is the initial amount of energy that needs to be invested to kickstart a chemical reaction or to nudge a chemical reaction into happening. If you look at this diagram, you can see that this reaction is exergonic. You have a high energy reactant being broken down into a low energy product. This chemical reaction is going to have a negative delta G. It's going to have a release in energy. You could determine the release in energy by looking at the difference between the reactants and products. Now, since this reaction is exergonic, we would also say that it's spontaneous. Even though it's spontaneous, it still needs this initial kickstart in order to get the chemical reaction to take place. The activation energy is usually found as thermal energy or as heat that gets absorbed from the surroundings. In other words, by having the reactants absorb heat from the surroundings, those reactants have increased kinetic energy. They begin moving faster. And this is going to be that initial nudge that gets that chemical reaction to take place. So what do enzymes actually do? Enzymes catalyze chemical reactions by lowering the activation energy. They reduce the amount of energy necessary to kickstart a chemical reaction. This means that enzymes allow chemical reactions to occur at lower temperatures. They occur at lower temperatures because the reactants need to absorb less thermal energy from their environment to get the reaction going. Notice that enzymes do not have any effect on delta G. They do not have any effect on the amount of energy released from a chemical reaction. They simply make them occur easier. How do enzymes physically work? An enzyme has an important region called the active site. The active site is the specific area of the enzyme that the substrate is going to bind to. Enzymes work through a lock and key mechanism. Every enzyme is extremely specific for the substrate it acts on. The active site is the lock, the substrate is the key. The shape of the active site is determined by the sequence of amino acids in that region. When the substrate enters the active site, it forms the enzyme-substrate complex. And this is where the enzyme is going to do its work. When the substrate enters the active site, you can see in this picture here that the enzyme actually slightly changes its shape. We call that induced fit, and that induced fit is going to put some strain on the reactant or some strain on the substrate, and it's going to begin facilitating that chemical reaction. Physically, enzymes catalyze chemical reactions, or enzymes lower the activation energy through two major mechanisms. First, if the job of the enzyme is to perform dehydration synthesis, if it's trying to make a chemical bond, it's going to make that chemical bond occur easier and faster by placing the substrates in the correct locations, by physically putting those two substrates together, allowing that dehydration synthesis to happen that much easier. If the job of the enzyme is to break a chemical bond through hydrolysis, that enzyme is going to help hydrolysis to occur easier by putting physical strain on the chemical bonds, by beginning to bend and contort that molecule to help those bonds break easier. These are the two major methods by which enzymes lower activation energy. Enzymes can also provide a favorable environment for the reaction to take place, or they could bond to the substrates themselves. Here we see what I'm talking about. Here we have the substrates enter the active site of the enzyme. Notice the enzyme's active site has a very specific shape that's unique to that substrate. While the substrate is in the active site, we have the enzyme-substrate complex. This is where the enzyme is going to speed up that chemical reaction and lower the activation energy by either orienting the substrates correctly or by putting strain on them, causing that chemical reaction to take place. And as always, our enzyme is not used up. Since enzymes are proteins, there are three major factors that will affect the functioning of an enzyme. You have temperature, you have pH, and you have salinity. Here we're going to look at temperature and pH. Every enzyme is going to operate at an optimal temperature and an optimal pH. If you look at all human enzymes operate at about 37 degrees Celsius, or body temperature. If you compare these enzymes to the enzymes found inside heat-tolerant bacteria, those enzymes operate at a much higher temperature. If you look at the enzymes within the human body, different enzymes operate at different pHs. The enzymes in the stomach, like pepsin operates at a pH of 2. Operates in a very acidic environment. If you look at trypsin, found in the small intestines, it operates at a very different pH, a pH of 8. What would happen if you change the pH? For example, what would happen if you took trypsin and put it into the stomach? It would cause that enzyme to denature. Those chemical bonds that hold it together would break, it would unravel, and it would be rendered useless. The same thing can take place by increasing temperature. As you increase temperature, the rate of reaction increases. This continues to happen until a critical temperature is met and the enzyme denatures. By reducing temperature, you would not denature the enzyme. Instead, you would simply just slow down the rate of chemical reaction. Sometimes there are factors that are bound to enzymes that they require in order to properly function. We call these things cofactors. Cofactors are non-protein enzyme helpers. They could be organic or they could be inorganic. If they are inorganic, they are not made up of carbon. It could be a metal like zinc functions as a cofactor. An organic cofactor contains carbon and we call them coenzymes. Most vitamins function as coenzymes. Many of the enzymes present in the body cannot properly function unless that vitamin is bound to it. This is why nutritional deficiencies, where a person does not obtain enough of a certain type of vitamin, could severely hinder the functioning of a region of their body. Those enzymes cannot properly function unless that vitamin is bound to the enzyme. Many things can alter the functioning of an enzyme. Here we examine inhibitors. Some examples of inhibitors are things like toxins, poisons, pesticides, or antibiotics that are taken into the body of an organism. There are two types of inhibitors. Both of these inhibitors are going to inhibit or stop the functioning of an enzyme. The first type of inhibitor is called a competitive inhibitor. The competitive inhibitor resembles the substrate. It looks like the substrate, so it's going to compete with the substrate for the active site. These competitive inhibitors are reversible. They're reversible because you can reverse the action of it by simply adding more substrate. If you add more substrate, the substrate will outcompete the inhibitor for the active site. The other type of inhibitor is a non-competitive inhibitor. With a non-competitive inhibitor, they bind to a different region of the enzyme. By binding to a different region of the enzyme, they change the shape of the active site. Once that active site has changed shape, the substrate will no longer be able to bind to it. These inhibitors are non-reversible. They are often deadly. A good example of a non-competitive inhibitor is penicillin. Penicillin is a type of antibiotic, and it binds to a key enzyme used in bacteria. The bacteria use this enzyme to manufacture their cell walls. By inhibiting that enzyme, bacteria cannot make new cell walls and die as an end result. Pesticides like DDT are non-competitive inhibitors. Different toxins and poisons like nerve gas, the technical name is sarin, is also a non-competitive inhibitor. How do different enzymes evolve over time? We know that the shape of an enzyme is determined by the sequence of amino acids. The sequence of amino acids is determined by the sequence in DNA. Well, often changes in DNA take place. We call these changes mutations. And when a mutation in DNA takes place, the sequence of DNA changes. If the sequence of DNA changes, the sequence of amino acids in the enzyme will change. If the enzyme changes shape, it's going to alter what type of substrate it can act on. If an enzyme changes its shape through a mutation, and that new change creates an active site that benefits the organism as a whole, that organism will be more likely to survive, that organism will be more likely to reproduce, and it will pass on this gene to its offspring. And its offspring will then contain this enzyme that now has this new and novel function to help it to survive. So you can see that through small changes in DNA called mutations, and through the process of natural selection, that new enzymes can evolve over time and help organisms adapt to their environment. We often think of inhibitors as being bad, but many times inhibitors can be good. The body of an organism needs to be able to, or we need to be able to, control all of our metabolic pathways. You don't want all of your metabolic pathways running all the time. You want to be able to turn them on and turn them off depending upon whether or not you need them. We know that every metabolic pathway contains many chemical reactions, and every chemical reaction is catalyzed by an enzyme. So it's very easy to turn on and turn off chemical reactions in the body by selectively turning off and turning on enzymes. What we'll see now is how we have the ability to control our enzymes, or how we have the ability to turn them on or turn them off, depending upon whether or not we want a metabolic pathway running. This form of regulation, this form of controlling our metabolic pathways and chemical reactions using our enzymes, is called allosteric regulation. Here we look at an allosteric enzyme. An allosteric enzyme is a type of enzyme that can be turned on or turned off. It has two forms. It has an active form where it's turned on, and it has an inactive form where it's turned off. Different chemicals are used within the body to turn on or turn off an allosteric enzyme. Activators are chemicals that lock the allosteric enzyme in the active formation. Inhibitors are going to be chemicals that lock the enzyme in the inactive form. You can see that when the enzyme changes from the active form to the inactive form, or vice versa, it actually undergoes a change in shape. The activators and inhibitors function by locking the allosteric enzyme into one of those different shapes. Allosteric regulation can also be used for cooperativity. Cooperativity is going to enhance the effect of an enzyme. If we look back at this picture here we see a substrate binding to the active site of an allosteric enzyme. Well, you can see that this allosteric enzyme has multiple active sites. In this case, it has 1, it has 2, it has 3, it has 4 active sites. When the substrate binds to the active site, it then locks it into its place. So once one substrate binds, 3 more substrates can bind to that allosteric enzyme. So the binding of one substrate causes the binding of more substrates, and this is able to amplify the response of the enzyme, and that's essentially what cooperativity is. Allosteric regulators, these inhibitors and activators, are pretty good drug candidates. When doctors are searching for ways to control chemical reactions in the body, they look for these allosteric regulators to prescribe as medicine. A good example involves an enzyme called a caspase. Here you see a picture of a caspase, and this is an enzyme that is found within the inflammatory response. So doctors were searching for a way to decrease inflammation. Well, what they did was they searched for an allosteric inhibitor. If you were to give the patient this allosteric inhibitor, it would bind to the caspase enzyme, lock it into its inactive form, and reduce the overall amount of inflammation. So we can see, based upon this example, that allosteric regulators are pretty good candidates for drugs. Allosteric regulation is going to be used in the process of feedback inhibition. If you remember, feedback inhibition is used to regulate your metabolic pathways. It's used as a form of control in order to maintain homeostasis. The definition of feedback inhibition is that the end product is going to be used to inhibit the first step in a metabolic pathway. Here we look at a particular metabolic pathway, and this is an anabolic pathway. It consumes energy, it's building something. And this metabolic pathway contains 5 chemical reactions. Since it has 5 chemical reactions, it utilizes 5 different enzymes. In this pathway, threonine is going to be converted into isoleucine. In order to do this, the organism needs to invest a lot of energy to build this isoleucine. Well, what happens if we have enough isoleucine? What happens if we don't want to produce any more of it? When isoleucine, the end product, builds up, isoleucine is going to act as an allosteric inhibitor. It binds to the first enzyme in the first chemical reaction. Binding of the end product to the initial enzyme is going to lock that initial enzyme, which is an allosteric enzyme, into the inactive form. And it's going to turn off this entire metabolic pathway. Well, when's this pathway going to turn back on? When we run out of isoleucine. When you run out of isoleucine, you no longer have your allosteric inhibitor, and the pathway turns back on leading to the production of more isoleucine. So you can see that the cell and an organism as a whole uses allosteric regulation, and it uses feedback inhibition. They really go hand in hand in order to regulate its metabolic pathways. Finally, when we look at cells, cells contain many different organelles. What's the purpose of having different organelles within the cell? What's the purpose of having a smooth and a rough endoplasmic reticulum, a Golgi apparatus, peroxisomes? What's the point of having different cellular structures? They isolate different enzymes. Different enzymes are found within different organelles. By isolating different enzymes, you isolate the different chemical reactions within a cell. So organelles are a way of localizing the different chemical reactions taking place within a cell. It's a way to have chemical reactions taking place in certain locations within that cell. Here you see a picture of a mitochondria, and you see the structure of a mitochondria. And it has the structure that it contains in order to isolate different chemical reactions in different locations. You isolate different chemical reactions by isolating those enzymes. That concludes this video.